So raise your hand if you know someone in your immediate family or circle of friends who suffers from some form of mental illness. Yeah, I thought so, not surprised. And raise your hand if you think that basic research on fruit flies has anything to do with understanding mental illness in humans. Yeah, I thought so. I'm also not surprised. I can see I got my work cut out for me here. As we heard from Dr. Insel this morning, psychiatric disorders like autism, depression, and schizophrenia take a terrible toll on human suffering. We know much less about their treatment and the understanding of their basic mechanisms than we do about diseases of the body. Think about it. In 2013, the second decade of the millennium, if you're concerned about a cancer diagnosis and you go to your doctor, you get bone scans, biopsies, and blood tests. In 2013, if you're concerned about a depression diagnosis, you go to your doctor and what do you get? A questionnaire. Part of the reason for this is that we have an oversimplified and increasingly outmoded view of the biological basis of psychiatric disorders. We tend to view them, and the popular press aids and abets this view, as chemical imbalances in the brain, as if the brain were some kind of bag of chemical soup full of dopamine, serotonin, and norepinephrine. This view is conditioned by the fact that many of the drugs that are prescribed to treat these disorders, like Prozac, act by globally changing brain chemistry, as if the brain were indeed a bag of chemical soup. But that can't be the answer because these drugs actually don't work all that well. A lot of people won't take them or stop taking them because of their unpleasant side effects. These drugs have so many side effects because using them to treat a complex psychiatric disorder is a bit like trying to change your engine oil by opening a can and pouring it all over the engine block. Some of it will dribble into the right place, but a lot of it will do more harm than good. Now, an emerging view that you also heard about from Dr. Insel this morning is that psychiatric disorders are actually disturbances of neural circuits that mediate emotion, mood, and affect. When we think about cognition, we analogize the brain to a computer. That's no problem. Well, it turns out that the computer analogy is just as valid for emotion. It's just that we don't tend to think about it that way. But we know much less about the circuit basis of psychiatric disorders because of the overwhelming dominance of this chemical imbalance hypothesis. Now, it's not that chemicals are not important in psychiatric disorders. It's just that they don't bathe the brain like soup. Rather, they're released in very specific locations, and they act on specific synapses to change the flow of information in the brain. So if we ever really want to understand the biological basis of psychiatric disorders, we need to pinpoint these locations in the brain where these chemicals act. Otherwise, we're going to keep pouring oil all over our mental engines and suffering the consequences. Now, to begin to overcome our ignorance of the role of brain chemistry and brain circuitry, it's helpful to work on what we biologists call model organisms. Animals like fruit flies and laboratory mice, in which we can apply powerful genetic techniques to molecularly identify and pinpoint specific classes of neurons, as you heard about in Alan Jones's talk this morning. Moreover, once we can do that, we can actually activate specific neurons, or we can destroy or inhibit the activity of those neurons. So if we inhibit a particular type of neuron and we find that a behavior is blocked, we can conclude that those neurons are necessary for that behavior. On the other hand, if we activate a group of neurons and we find that that produces the behavior, we can conclude that those neurons are sufficient for the behavior. So in this way, by doing this kind of test, we can draw cause and effect relationships between the activity of specific neurons in particular circuits and particular behaviors, something that is extremely difficult, if not impossible, to do right now in humans. But can an organism like a fruit fly, it's a great model organism because it's got a small brain, it's capable of complex and sophisticated behaviors, it breeds quickly, and it's cheap. But can an organism like this teach us anything about emotion-like states? Do these organisms even have emotion-like states, or are they just little digital robots? Charles Darwin believed that insects had emotion and expressed them in their behaviors, as he wrote in his 1872 monograph on the expression of the emotions in man and animals. And my eponymous colleague Seymour Benzer believed it as well. Seymour is the man that introduced the use of Drosophila here at Caltech in the 60s as a model organism to study the connection between genes and behavior. Seymour recruited me to Caltech in the late 1980s. He was my Jedi and my rabbi while he was here. And Seymour taught me both to love flies and also to play with science. So how do we ask this question? It's one thing to believe that flies have emotion-like states, but how do we actually find out whether that's true or not? Now, in humans, we often infer emotional states, as you'll hear later today, from facial expressions. However, it's a little difficult to do that in fruit flies. It's kind of like landing on Mars and looking out the window of your spaceship at all the little green men who are surrounding it and trying to figure out, how do I find out if they have emotions or not? What can we do? It's not so easy. Well, one of the ways that we can start is to try to come up with some general characteristics or properties of emotion-like states, such as arousal, and see if we can identify any fly behaviors that might exhibit some of those properties. So 3 important ones that I can think of are persistence, gradations in intensity, and valence. Persistence means long-lasting. We all know that the stimulus that triggers an emotion causes that emotion to last long after the stimulus is gone. Gradations of intensity means what it sounds like. You can dial up the intensity or dial down the intensity of an emotion. If you're a little bit unhappy, the corners of your mouth turn down and you sniffle, and if you're very unhappy, tears pour down your face and you might sob. Valence means good or bad, positive or negative. So we decided to see if flies could be provoked into showing the kind of behavior that you see by the proverbial wasp at the picnic table. You know, the one that keeps coming back to your hamburger the more vigorously you try to swat it away and seems to keep getting irritated. So we built a device which we call a puffomat, in which we could deliver little brief air puffs to fruit flies in these plastic tubes on our laboratory bench and blow them away. And what we found is that if we gave these flies in the puffomat several puffs in a row, they became somewhat hyperactive and continued to run around for some time after the air puffs actually stopped and took a while to calm down. So we quantified this behavior using custom locomotor tracking software developed with my collaborator Pietro Perona, who's in the electrical engineering division here at Caltech. And what this quantification showed us is that upon experiencing a train of these air puffs, the flies appear to enter a kind of state of hyperactivity, which is persistent, long-lasting, and also appears to be graded. More puffs or more intense puffs make the state last for a longer period of time. So now we wanted to try to understand something about what controls the duration of this state. So we decided to use our puffomat and our automated tracking software to screen through hundreds of lines of mutant fruit flies to see if we could find any that showed abnormal responses to the air puffs. And this is one of the great things about fruit flies. There are repositories where you can just pick up the phone and order hundreds of vials of flies of different mutants and screen them in your assay and then find out what gene is affected in the mutation. So doing this screen, we discovered one mutant that took much longer than normal to calm down after the air puffs. And when we examined the gene that was affected in this mutation, it turned out to encode a dopamine receptor. That's right, flies, like people, have dopamine, and it acts on their brains and on their synapses through the same dopamine receptor molecules that you and I have. Dopamine plays a number of important functions in the brain, including in attention, Arousal, reward, and disorders of the dopamine system have been linked to a number of mental disorders, including drug abuse, Parkinson's disease, and ADHD. Now, in genetics, it's a little counterintuitive. We tend to infer the normal function of something by what doesn't happen when we take it away, by the opposite of what we see when we take it away. So when we take away the dopamine receptor and the flies take longer to calm down, from that we infer that the normal function of this receptor and dopamine is to cause the flies to calm down faster after the puff. And that's a bit reminiscent of ADHD, which has been linked to disorders of the dopamine system in humans. Indeed, if we increase the levels of dopamine in normal flies by feeding them cocaine after getting the appropriate DEA license, oh my God. We find indeed that these cocaine-fed flies calm down faster than normal flies do. And that's also reminiscent of ADHD, which is often treated with drugs like Ritalin that act similarly to cocaine. So slowly I began to realize that what started out as a rather playful attempt to try to annoy fruit flies might actually have some relevance to a human psychiatric disorder. Now, how far does this analogy go? As many of you know, individuals afflicted with ADHD also have learning disabilities. Is that true of our dopamine receptor mutant flies? Remarkably, the answer is yes. As Seymour showed back in the 1970s, flies, like songbirds, as you just heard, are capable of learning. You can train a fly to avoid an odor, shown here in blue, if you pair that odor with a shock. Then when you give those trained flies the chance to choose between a tube with the shock-paired odor and another odor, it avoids the tube containing the blue odor that was paired with shock. Well, if you do this test on dopamine receptor mutant flies, they don't learn. Their learning score is 0. They flunk out of Caltech. Now, so that means that these flies have two abnormalities, or phenotypes, as we geneticists call them, that one finds in ADHD: hyperactivity and learning disability. Now, what's the causal relationship, if anything, between these phenotypes? In ADHD, it's often assumed that the hyperactivity causes the learning disability. The kids can't sit still long enough to focus, so they don't learn. But it could equally be the case that it's the learning disabilities that cause the hyperactivity. Because the kids can't learn, they look for other things to distract their attention. And a final possibility is that there's no relationship at all between learning disabilities and hyperactivity, but that they are caused by a common underlying mechanism in ADHD. Now, people have been wondering about this for a long time in humans, but in flies we can actually test this. And the way that we do this is to delve deeply into the mind of the fly and begin to untangle its circuitry using genetics. We take our dopamine receptor mutant flies and we genetically restore or cure the dopamine receptor by putting a good copy of the dopamine receptor gene back into the fly brain. But in each fly, we put it back only into certain neurons and not in others. And then we test each of these flies for their ability to learn and for hyperactivity. Remarkably, we find we can completely dissociate these two abnormalities. If we put a good copy of the dopamine receptor back in this elliptical structure called the central complex, the flies are no longer hyperactive, but they still can't learn. On the other hand, if we put the receptor back in a different structure called the mushroom body, the learning deficit is rescued, the flies learn well, but they're still hyperactive. What that tells us is that dopamine is not bathing the brain of these flies like soup. Rather, it's acting to control two different functions on two different circuits. So the reason there are two things wrong with our dopamine receptor fly is that the same receptor is controlling two different functions in two different regions of the brain. Whether the same thing is true in ADHD in humans, we don't know, but these kinds of results should at least cause us to consider that possibility. So these results make me and my colleagues more convinced than ever that the brain is not a bag of chemical soup, and it's a mistake to try to treat complex psychiatric disorders just by changing the flavor of the soup. What we need to do is to use our ingenuity and our scientific knowledge to try to design a new generation of treatments that are targeted to specific neurons and specific regions of the brain that are affected in particular psychiatric disorders. If we can do that, we may be able to cure these disorders without the unpleasant side effects, putting the oil back in our mental engines just where it's needed. Thank you very much.